All right, I think we're live. Welcome everybody. Let me quickly activate the Twitter stream as well and the YouTube stream. And then welcome for the first time in a while on screen, Professor Astrid Prinz, who is going to be our guest today. Astrid did her PhD in biochemistry at the Technical University in Munich and at the Max Planck Institute in Martinsried, and back then worked on geometrically simple neuronal networks that she laid out herself from snails and looked at action potential propagation in these networks. I met Astrid when she was a postdoc in Eve Marder's lab. She started in 2000. I started my PhD shortly after. And she spent 4 years in Eve's lab at Brandeis University in Boston and published back then what is now a really famous and beautiful paper still on the ability of neurons and neural networks to find multiple equally valid solutions for the same problem, looking at, for example, ion channel distributions in single neurons, or looking at network configurations. And she used an approach that I think is still with her that she charmingly called the hammer approach. So back when I saw her practice talks, for example, for her professorship that she took in 2005 at Emory University, she always started out with a hammer because unlike the modern machine learning methods that are looking for a single solution, she simply calculated all of the solution, or she swept vast parameter spaces to look at all the different combinations of parameter instantiations to come up with the same solution. And I think we'll hear again about work like this from Astrid today. I think she has still been working, or still is working, on the stomatogastric ganglion of lobster, but she has since then also veered into other animals or into other systems. So she's looking at fly neural systems or fly larvae systems, as well as looking at homeostasis specifically in mice after spinal cord injury, and maybe we'll hear a little bit about other systems as well. Astrid, thank you very much for coming. The floor is yours. Just very quickly, for those of you who are new in the audience, ask your questions via the question box or say hello in the chat section. And at the end of the talk, I will invite you to come to the podium, so to speak, by joining us in a separate Zoom room if you want to say hi to Astrid. Please feel free. So without further ado, Astrid, I will maximize your screen, and I'm looking forward to your talk.
Okay, thank you, Tim, for the nice introduction and also for inviting me to this very exciting forum. As you told me earlier, this seems to be a pandemic baby, and we don't know what the future of it will be, but I think it's very impressive how you're pulling together all these exciting speakers for these online seminars. One minor point, my PhD was at the Max Planck Institute for Biochemistry, but it was actually in physics. So that might explain why I may not have necessarily biochemistry answers for some questions that might come up in the context of this. I'm really a physicist by training. Okay, so I will talk today about neural circuit parameter variability, robustness, and homeostasis. And so this all goes back to this, I think, very succinctly stated observation by Walter Cannon, a famous physiologist in the early 20th century. Somehow, the unstable stuff of which we are composed has learned the trick of maintaining stability. And I think the work that I'm going to tell you about today, even though it goes back all the way to my time as a postdoc in Eve Marder's lab, is only really starting to scratch the surface about regarding how the unstable stuff that makes us up at the cellular level and at the subcellular level might actually be achieving stability. Okay, my next slide is just a brief outline. I will jump right into introducing you to the stomatogastric ganglion as a model system, because I think that some of you may not be familiar with that. I'll talk about parameter variability in this system, and then the notion of a solution space when we think about how this and other neural systems can achieve their biological function. And then as a special feature of these solution spaces, I'll talk a little bit about parameter correlations, linear relationships between parameters that are emerging in the system, and then try to wrap it up and talk about what all of this might mean for how neural systems can homeostatically regulate to maintain proper function. So the crustacean stomatogastric ganglion, or I'm sure I'll fall into lingo and call it STG now and then. It is a ganglion, which means a clump of nerve cells, only about 30 of them, that is part of the stomatogastric nervous system that's shown in yellow here, wrapped around the crustacean stomach. So crustaceans, we're talking about lobsters or crabs here. And this nervous system its major function is to generate rhythmic electrical activity that governs rhythmic movements of the stomach. There are two different rhythms. There is a gastric rhythm that moves teeth inside the lobster stomach that grind up food. And then there is what we'll be mostly talking about today. There is the pyloric system, which is moving a filtering system in the pylorus that filters food as it passes through the stomach and toward the later digestive system. You can dissect this nervous system off of the stomach and place it in a dish where schematically it looks like this, and you can then intracellularly record from neurons in the stomatogastric ganglion where you observe rhythmic bursting activity. And less invasively, you can also record those same bursts as they're traveling down motor nerves when they would normally then send their activity to muscles in order to move the stomach around. It's not advancing. Now it is. Okay. So a little bit more about this pyloric pattern generator. It can basically be boiled down to three different types of neurons. They are abbreviated. You don't need to worry about the names here. There's a lateral pyloric neuron, there are pyloric neurons, and there is an anterior burster and a pyloric dilator neuron. The essence of this is that these are mutually inhibitory neurons. So these synapses are all inhibitory, and at the core of it is this pacemaker kernel, AB/PD, anterior burster/pyloric dilator, that intrinsically, even in isolation from any other cells, is producing this rhythmic bursting pattern with about a 1 to 2 second period. And the other types of neurons are then also producing a rhythmic pattern, but only in response to inhibition, in rebound from inhibition that they receive from the pacemaker kernel. So you end up with a triphasic rhythmic motor pattern that will move selective groups of muscles in the stomach to produce this food filtering. So a question that many have, maybe especially in a forum that involves neurotheorists, is why would you study a system like that? And if my slides advance, there seems to be a bit of a delay with slide advancing. I clicked the button to advance slides, but it's giving me the hourglass right now, so I'll just talk about what's on the next slide, and then it'll pop up, I hope. So the next slide is kind of laying out why do we study the crustacean stomatogastric ganglion. So one point is, now it advanced, okay. One point is that like many other invertebrate nervous systems, it consists of so-called identified neurons. So what does that mean? It means that we have, and down here is a reminder of the circuit, we have in many cases just one copy of a given neuron type in every animal. So for example, there's only one lateral pyloric neuron in every crab's stomatogastric nervous system. So that makes it much more controllable than many vertebrate brain structures where you have thousands or tens of thousands of neurons of a given type that you then need fancy optogenetic techniques to be able to control the whole population. These identified neurons also always make synapses with the same synaptic partners. They produce the same electrical activity in every animal, and they have the same projections to muscles. So it's a very defined, highly defined structure, and you can go in experimentally and locate this particular neuron, let's say the LP neuron again, in a crab and study it and do that in 20 crabs and have very high reproducibility and very easy experimental access. The output pattern is also very stereotyped, which means that we can relatively easily assess, is this thing doing its job? Is it producing the right electrical activity or not? Which again, maybe for a cortical column, for example, might be a tougher decision to make. What you need to know what it's supposed to be doing in order to judge whether it's doing the right job. And it's also doing producing its activity in clearly defined functional ranges, so we know how far can you change the temperature until all of this breaks down. And so this is overall a very experimentally tractable and highly defined system, but nonetheless, I hope I'll be able to convince you, over the past decades we have derived many generalizable insights from this simple system that apply to pattern generating systems and also neural systems in general. Advancing my slide, this time it went quickly. Okay, so that kind of gave you a little bit of an introduction to the system that we're dealing with here. So now in this system, it has been known for quite a while that there is, despite this stereotyped nature of the output that is being generated and the very stereotyped nature of the neurons, of these identified neurons, despite all of that, there is quite a bit of parameter variability. And here's just a figure from older work from Jorge Golowasz when he was working with Yves Marder, where he went into a pyloric pacemaker neuron and or another pyloric neuron of a different type and measured the maximal conductance for different types of membrane currents in these neurons. So these three currents shown here, the A, the KCA, and the KD, these are three different types of potassium currents for which there are channels in the membrane of these neurons. And every data point here is from the same identified neuron, but in different animals. And you see that even though these neurons produce this very stereotyped activity, they do so on the basis of very different underlying parameters. You have variability here. For example, here, this calcium-dependent potassium conductance varies about fourfold between different animals. So you have highly reliable and stereotyped electrical activity produced by this circuit, but on the basis of highly variable cellular properties. And I can tell you now, and you'll see later, that synaptic properties are also highly variable in this circuit. So to kind of understand how that can be, how can you produce such reliable activity on the basis of such variable properties, we turn to modeling, and for this forum I don't have to explain too much about what a model neuron is. In our particular case, we were starting with a single compartment model neuron that contains 8 different membrane conductances including sodium, calcium, and potassium, and mixed and leak conductances, and a calcium buffer model. And just as a reminder, this then makes this a conductance-based model, and many here will probably be familiar with what a, now I'm losing my mouse. Can anybody still see my cursor? I don't think so. There you go, okay.
We can see your cursor.
Okay, now I can see it too. I had lost it for a moment. Technical glitches in pandemic times. We've all learned to deal with it, I guess. So just as a reminder, we're talking here about a conductance-based model. So what that really is in the computer a collection of differential equations and other equations that describe, for each of these 8 membrane currents, describe how the current depends on the membrane potential. And there are some equations to describe the calcium dynamics in the cell, and then it all gets tied together by this master equation that describes how the membrane potential is changing as a function of the currents. So that is all I want to say about the details of this model. So it is a single compartment conductance-based model with 8 conductances. And we decided to use what Tim already alluded to, and that is what I used to symbolize with a hammer, because I think of this as a brute force approach. We basically took this approach that we said we're completely agnostic as to how the electrical activity and output of the neuron depend on the underlying properties, in this case the 8 membrane conductances. And so we'll just explore it exhaustively. So we're looking here at an 8-dimensional conductance space, of which I can obviously only show 3 on this 2-dimensional slide. And these parameters, these 8 parameters that we chose to explore, are the maximal conductances for these 8 membrane currents. And we covered this 8-dimensional parameter space with a grid of simulation points where each conductance could vary from 0 to some physiologically reasonable maximum in equal increments. And then we just programmed the computer to simulate all possible combinations of these parameter values and determine what does the model do when it has this particular parameter combination. So we chose 6 different values for each conductance. That gives us 6 to the 8th, about 1.7 million motor neurons, model neurons. Advancing the slide. The next slide will show, once it advances, that when you look at different parameter combinations on the right here, these bar graphs just indicate what is the value of each of these maximal conductances. When you simulate what they do, you find a variety of different activity patterns, from silence to regular spiking to spiking without a sodium conductance, so that would be calcium spiking. And then you find a lot of different burst patterns, which is what we were hoping for, since we were trying to model bursting stomatogastric neurons, and some other maybe slightly more exotic types of behavior. So all of this resides in a database that basically relates the membrane parameters, the maximal conductances, to the output pattern. And this database is now, over 15 years old, but we're still using it actually on a regular basis. And one of the first things that we did with this was to just go into this database and mine it to pull out all the models that were producing bursting activity within the functional range that we observe in living stomatogastric stomatogastric neurons. And when I say within the functional range, I mean in terms of burst period, burst duration, number of spikes per burst, and a variety of other characteristics of the bursting when you do that, you can whittle down this database of 1.7 million to a small subset that really match the biological activity in all criteria. And here's an example of 9 of these models that were pulled out of the database, and we're looking here at their maximal conductances for 5 of their 8 different membrane currents, actual value in the database, and then standard deviation over mean, or coefficient of variation here. And you find that just like in the experimental data, which I've repeated down here, so we saw this plot before, just like in the experimental data, you find ranges of these parameters, even though all 9 models produce very similar and biologically meaningful electrical activity. And so you see, for example, for the sodium conductance here, it varies about 4-fold from 100 to 400. So the bottom line then is that at the cellular level, both experiments and modeling show that you can produce very similar and biologically functional electrical activity on the basis of variable parameters. So that by now, I think, is widely accepted. It's consistent between experiments and models. We then went on to apply this to the network level, and this is the paper that Tim was referring to. Again, I'm waiting for my slide to pop up. So the next slide will show, as a reminder, what network are we talking about. We're talking about the pyloric circuit in the stomatogastric ganglion. Here is again its general layout. And we applied the same approach in the sense that we did a brute force parameter exploration, applying the hammer to this thing, and doing that by plugging 5 different pacemaker model versions into this position of the network, 5 different lateral pyloric model versions, 6 different pyloric versions, and then for each of the inhibitory synapses in the circuit, we also scan through 5 to 6 different values of synaptic strength, actually on a semi-logarithmic scale, so really spanning several orders of magnitude, and again did all possible combinations, 20 million in this case, to produce a now database of network models. And you find the same phenomena that you found at the network level, that is, different models in this database that differ in their, so at the top row here we have 5 examples that have the same model neurons in the 3 positions but differ in their synaptic strengths, and in the bottom row we have 5 examples that have the same synaptic strengths but differ in their cellular properties. And you find again very different behaviors at the network level, but including behaviors that look like, and the 2 examples at the right here are examples of that. You do find network models in the database that produce physiologically meaningful activity now in really in 15 different characteristics of the rhythm, including period, burst duration of each of the members of the network, phase relationships between them, duty cycles. So a lot of criteria for each of which we require a model network to be in the physiologically observed range, and only then do we call it a proper pyloric network model. When you do that, you find about 2% of this database to be proper pyloric network models, but 2% of 20 million is about 400,000. So that is still a very high number that we can then mine for their properties as a population. And here are just 2 examples of them. So we have at the top the simulated electrical activity of the 3 neurons in the network for 2 examples, and at the bottom you see some of their cellular properties and some of their synaptic properties. And if you compare left and right, now I have to look at my other screen because this is too small on this screen. for example, here, the pacemaker neuron has a relatively large, slow calcium conductance, whereas in this other network, that same conductance is relatively small. Or at the synaptic level here, you have a large synapse, a strong synapse from the pacemaker to the lateral pyloric neuron, whereas over here, it's pretty small. And yet, these two networks produce highly stereotyped and very similar electrical activity. So that is really the main message here out of these two databases at the cellular and network level, that you can get functional electrical activity on the basis of different variable underlying properties, and properties includes cellular properties and synaptic properties. And to me, this then kind of inspired this, what I discovered in chemistry when they analyze chemical networks is called ensemble modeling. And so I'm advocating for adopting that term here also, this idea that rather than modeling a neural system with one specific model, with one specific parameter set, maybe we should kind of embrace the biological variability and create entire ensembles of models that vary in their properties, in their underlying parameters, but produce the same activity. And I think one advantage of taking that ensemble modeling approach is that you might be less in danger of studying an idiosyncratic property of one particular parameter combination, because you can now analyze a whole population and see what are kind of general statements that you can make about the relationship between parameters and function, rather than falling into the trap of maybe studying kind of an exotic outlier. So I think ensemble modeling has a lot of potential in that respect, and it is being done by many people. So I should fully acknowledge that. One last thing I would like to point out here, maybe as a little bit of a jab toward the connectome, what this result down here shows is that yes, synapses are important, and figuring out who is connected to whom is very important. And Eve Marder said it is necessary for us to understand what's going on, but it may not be sufficient. And that is because, in a sense, these are two very different connectomes, right? The synaptic connections are very different here, and so it's not that there's one recipe, like one connectome that an animal has to implement in order to produce its proper function. So I would caution that connectomes are absolutely necessary for us to know, but may not be sufficient to understand what is happening. And overall, this whole observation that you can get similar output from different underlying properties, I've always liked to call it parameter non-uniqueness. There is not one unique value for every parameter that produces proper function, because this is an old version of this slide, degeneracy is crossed out here because I just didn't find the term very appealing. But I now realize that this might be a lost cause. I think degeneracy is the term that is being used in the field to describe the phenomenon that similar parameters lead to different activities. So, all right, let's call it degeneracy. Okay, so we see parameter variability at multiple levels in these neural systems, and that kind of raises the question, well, now that I have a list of parameter combinations that produce proper activity, how are they situated in parameter space, right? In this case, I'm exploring 8 parameters. There are, of course, many other, in the case of the single neuron even, there are, of course, many other parameters that are being held fixed in this analysis, but already an 8-dimensional parameter space is, at least for me, impossible to visualize. And so the question is, how can we understand where these parameters that produce functional activity are located in parameter space? And that gets us to the notion of a solution space. So when we first started doing this kind of exhaustive analysis, it was not at all clear what solution spaces would look like. So again, I'm defining the solution space as the subspace of a larger parameter space where the parameter combinations in that subspace produce functional activity. And so at first, a priori, it's not clear what the structure of these solution spaces should probably is. You know, we don't know if it's one nice convex connected region, or if it's several smaller regions, or even little pockets scattered around parameter space, or if it's a connected but not necessarily convex structure. And I will give away what is no longer a secret, that at least in our work and in many other people's work, what we find is that these solution spaces seem to usually be connected, but not necessarily of simple shapes. So this is probably the most fitting of these four cartoon images. But how do we know that? How do we visualize these things, and how do we figure out how they are placed in parameter space? So to do that, we developed together with the lab of Tim Hickey, a computer scientist at Brandeis, we developed a visualization method that could visualize these high-dimensional data sets, these high-dimensional databases, and that is called dimensional stacking. So this slide, I hope I won't have to spend too much time on it and take you through it quickly. This is kind of the basic idea of dimensional stacking. So if you have, let's say you have a system that has only two parameters, to visualize it, you would simply plot a two-dimensional graph where you would, and if this two-dimensional space is covered by a grid like we have in these databases, then you could just color each pixel by whatever output feature of interest you want to look at. So you could make slow periods blue, make fast periods red, and basically look at how are these output features distributed in this parameter space, and especially where are the parameters that produce good functional activity. So 2 dimensions is easy, right? So this dimensional stacking idea is basically now stacking little plots like this where 2 parameters are systematically varied. In this case, the calcium-dependent potassium and the sodium conductance. You stack little plots like that into the next level of 2-dimensional organization, where within each of these little squares you vary these 2 parameters, but then you use 2 additional parameters like the delayed rectifier current here, Kd, and the transient calcium current to organize the dimensional stack at the next level. And since we're dealing here with an 8-dimensional parameter space for the single model database, and even more parameters for other newer databases that we've generated, you would, for this 8-dimensional space, you would have to do this 3 times. So you stack these red guys into these guys, and then these into the next level organized by 2 more parameters, and then stack those into the last level organized by the 2 remaining parameters. And so now you have an arrangement here where you have a dimensional stack that is organized by these different parameters, but that where every single entry in the database, every one of these 1.7 million entries is represented by its own pixel. So the entire database is there, but it's organized into a 2-dimensional display. So what does that look like? So here's a dimensional stack, and it's actually the only one I'm going to show in this talk, but there's a lot to see here. So what we're looking at here is the dimensional stack. Down here, these little bars indicate which conductance parameter organizes which level of this stack, both vertically and horizontally. So here the calcium-dependent potassium organizes the highest level of the stack, so it goes from 0 to 1 to 2 to 3 to 4 to 5 increments of its value, and so on for these lower levels of organization. And then we have color coded here just the general behavior of the neurons from the database. So gray indicates that the model with those parameters is silent, it doesn't produce spikes. Blue indicates tonic spiking, and green through red indicates bursting with various numbers of spikes per burst. And if you then use this color code to color each pixel according to its location in this two-dimensional stack, this is what you get. So you see that there's a lot of structure there, and that is the point of these dimensional stacks, and we've really learned a lot from them. So one thing you can already see is that if your calcium-dependent potassium conductance is 0, meaning you're in this column here, and your and your sodium conductance is 0, meaning you're in this row here, this entire square, almost this entire square, is gray. So if those two things are 0, you're not going to be so good at making action potentials. That's one feature I'm pointing out. And then if your calcium-dependent potassium conductance is 0, you're quite likely to be either silent or a tonically spiking neuron. Not many other activities going on here. And then another interesting feature is within each of these highest level squares, you see a kind of a progression from green to red up this diagonal here. And that means a progression from few spikes to many spikes per burst, but all of these being bursters. And that tells you that, so now you can see what organizes each of these highest level squares. That's the second conductance down. So that's the transient calcium conductance organizes this horizontally, and the delayed rectifier potassium conductance organizes this vertically. And so you see, as the transient calcium decreases going to the left, and the delayed rectifier increases, that is going up, as you're moving in this direction, you're going to get more and more spikes per burst. So you see how this dimensional stacking can be used to kind of learn something about the structure of these parameter spaces, and also of these solution spaces. Like if I now said, well, I am looking for a solution to the problem of producing, let's say, bursting activity with 3 spikes per burst, then I'm looking at yellow here. So I can look at where are the yellow solutions located, and that is basically the solution space for 3 spike bursting. And I can now look at my legend and kind of understand how this depends on the underlying parameters. So this has been a very helpful tool, and I could talk much more about what else we did with this dimensional stacking. Of course, it's most helpful because we have created these highly structured databases that do an exhaustive exploration. People have tried with some success to apply dimensional stacking also to less regular coverages of parameter space, for example, obtained by evolutionary algorithms. And I can talk offline about that, but that is really not the topic of today. Okay, so going back. So you see a lot of structure in here, including you see that there seems to be some correlation going on for this yellow solution space between this conductance that organizes the second level horizontally and this vertical one. And that gets us to this general observation that the next part of the talk will be about, and that is that within these solution spaces, we often find linear parameter correlations. well, we find parameter correlations in general, but many of them are linear. And that is reminiscent of experimental data that I have to explain here, because it kind of ties everything back to biology. So what we're looking at here is we're still in the stomatogastric world, stomatogastric ganglia, and we're looking here at cell type specific ion channel correlations. And this slide has a lot of information, so let me spend some time on it. First, on the left here, this is data, again, from the work of Jorge Golowash, who inappropriately is not cited here. He should really be cited here. We're looking at PD neurons, so that's one of those pacemaker neurons in the stomatogastric ganglion, and we're looking at two of its membrane conductances. That there's the A current, which is a potassium current, and then there is the H current, which is the hyperpolarization activated cation current that some of you may be familiar with. So another membrane current in these neurons. Each dot is an animal, and what we're looking at is electrophysiology data. So this was measured in individual neurons by voltage clamping them and determining the magnitude of these conductances. And you see that across this population of animals, you see a correlation between these two parameters. So at the electrophysiology level, you see that these two membrane conductances each vary widely, as we saw before, several fold on the horizontal axis, several fold on the vertical axis, but they don't vary independently. They seem to have a relationship across the population. On the right, you also see a lot of linear relationships, but these are not from electrophysiology. These are from, I'm keeping an eye on the clock and I really need to speed up a little bit. These are not from electrophysiology. These are from single-cell mRNA measurements of the messenger RNA coding for ion channels. And you have each little plot here is for a pair of different ion channels. So shell here is coding for a particular potassium channel. This on the horizontal axis here is coding for the same hyperpolarization activated channel. So each plot looks at a different pair of membrane conductances, of the mRNA for different membrane conductances. And each color here is for a different type of stomatogastric neuron. So you see there's LP, which we already encountered before, is blue everywhere. PD, one of the pacemakers, is black, and then there are some other cell types that we haven't encountered yet. And then you see that you see a lot of linear relationships, and I have to explain this data set. All these mRNAs were measured in all the cell types, and so there should be many more colors, like all the colors should be in every plot here. But they're not, because Dave Schultz, who created this beautiful data, is only plotting the data where he does find a linear relationship. And so there's a lot of information in here. Again, you do find between now the mRNAs coding for different types of ion channels a lot of linear relationships. These linear relationships are all positive. This is a point that I'll return to later. So when one mRNA is large, copy number is large, the other is also large. No negative relationships here. Second, you see for a given pair of membrane conductances, you can see linear relationships of different slope, different ratios, depending on the cell type. And then some cell types have a relationship between, let's say, this potassium current and this other potassium current. So GM, LP, and LG do seem to have a linear relationship between these two, although of different slope, but the other cell types do not have a linear relationship between these two. So what's emerging is that what is defining these cell types, and again, these are identified neurons, so they are highly specific in terms of what they do, who they connect with synaptically. These are very distinct cell types. What defines them is not that a PD needs to have X copy number of this channel and Y copy number of this other channel and needs to have exactly that value. But what defines a cell type seems to be a set of correlation rules. And different cell types have different correlations that they seem to be implementing across a population. Here, just like over here, every data point is a single cell data point from one animal. So we're looking at many animals here. So this is a lot to digest, but the bottom line here, if that's the only thing you take away, is that within these identified neurons, we find many linear relationships between ion channels, both at the mRNA level and at the electrophysiology level. And what seems to be defining a cell type is a different set of correlation rules. So this is very exciting, I think, and it made us go back into our single cell database. This is work by my former graduate student, Amber Hudson, now Amber Willett. And what she did is she took this whole database of 1.7 million models and she said, let me break it down into the model equivalent of a cell type. So how did she do that? She first broke the database down by just its overall activity, spiking models, silent, bursting models. And then she further broke those down into periodically or irregularly spiking or bursting. And then she broke the periodically spiking or bursting ones down in many different ways, but showing here, I'm just showing here two examples. One is she grouped them into categories by spike frequency in Hertz, or by, in the case of the bursters, by duty cycle, that is, which fraction of the period is taken up by the burst. And so she artificially generated subpopulations that we think are kind of the model equivalent of a cell type, because they're defined by their type of activity. And then she asked, within this database, within this cell type, for example, the spiking spiking models with a frequency between 25 and 50 hertz, do I see any linear relationships between their conductances, between their maximal conductance parameters? And the number of correlations that she found for each of these artificial cell types is given here. And so the bottom line is here, if you carve out artificial cell types, the equivalent of a cell type from a model database, you do find linear correlations, just like you see in the experiment. I will quickly go through this slide. This is just an example of this. So here we're again looking at the spiking models, and each panel here corresponds to a population with a different frequency out of the model database. And we're looking here at two parameters, that is the transient calcium conductance and the sodium conductance. And these heat maps here show the number of models in the database that perform this activity and have this particular combination of values of these two conductances. And you see emerging here what looks like kind of ridges that look like linear relationships that seem to be different in slope depending on the spiking frequency of the population. And to us, that was very reminiscent of the mRNA data that I just showed you earlier, where different cell types can show linear relationships, but of different slope. I have a noisy leaf blower outside. I'm going to close my door.
We can't hear it, so it's fine.
Okay, well, I could hear it. Now it's now the balcony is closed. Okay, sorry for the interruption. So The next slide is so complicated that I'm going to only give you the basic message, and I'll be happy to return to it in the interest of time. If you look at correlations in model populations that you make more and more refined, not only spiking at a certain frequency, but also with a certain spike amplitude or whatever you want to throw at it, you find that the correlations that you find in the population change as you throw additional constraints at it. And my main conclusion here is it's complicated. I have, even 10 years after this was published, I have still not wrapped my head around exactly what is going on here. So these solution spaces have pretty complex structures, depending on what activity you're looking for a solution for. I'm waiting for my slide to advance again. Okay, here is a last point in that regard about these correlations, and that is work from my most recent graduate student, Quinn Tian, that we're currently getting ready to publish. And what she basically found is that if she had a whole different database of Pyloric network models that she generated not by brute force hammer grid search, but by evolutionary algorithms that she used to generate a model population. And she found that if she pulls out of that, of her database, models that fall within the mean plus minus 2 standard deviations of the biologically observed network period. So this is, the black is biological data. She gets, she pulls out a certain population of models. If she makes the constraint tighter and she says, what if I want the period to be within plus minus 1 standard deviation of the biological mean, what population do I get then? And she found that she gets different populations, and in general she finds that any linear correlations that she observes with the looser constraint will tighten up with the tighter constraint, with the narrower constraint, and that is shown here in terms of the coefficient of the correlation coefficient, where the red population, which comes from the tighter constraint on the period, has these flanks of larger positive and negative values of the correlation coefficient. That her kind of conclusion here, but I put it with a question mark because we still need to work a bit on that, is that if you want your neuron or your neural system to perform a certain activity and you constrain that very loosely, let's say you only say, I need bursting, I don't care about the details of it, you might find that in your parameter space, different types of activity fall into whole different large regions, bursting maybe over here and spiking over here and so on. If you and her contention is, again, we need to verify that more carefully. If you then tighten your constraint, so you're moving to the left on this axis here, you might find that you go from these loosely defined regions to more like slices that maybe look a little bit like what we saw in the dimensional stack with that yellow region in there. And if you go tighter and tighter, you might get linear relationships that get tighter and tighter, and eventually, if you highly constrain your expected activity, then maybe you end up with only one or very few tightly situated parameter sets that can actually perform that activity. Okay, so we have seen that solution spaces exist. I did not actually explicitly show you, but we do find them to be continuous in most cases. They are structured around linear parameter relationships, in our case, mostly between membrane conductances. And so now the question is, how does a biological system navigate this kind of situation? You know, if the job of the neural system is to create a certain type of activity, that means it needs to be inside that solution space. How does it make sure it gets there in the first place during development? And how does it make sure it stays there even in the face of perturbations that might kick it out of there? How could it homeostatically regulate itself to return back to the solution space? Tim, I realize it's almost 10 o'clock. I'm almost done.
It's all right.
You said I can go a little bit over. Yeah, okay.
We're not hemorrhaging audience members at any rate.
We're not?
No. So I
okay,
think
good.
5 minutes if you want.
Stay with me, only a few more slides. One idea, so we know from experimental data and also from modeling work that there are multiple mechanisms in this and in other neural systems in place to homeostatically regulate neural activity. And I will only talk about a very narrow subset of them because of time and because that's what I can say the most about. And that is activity-dependent homeostatic regulation. There also are many mechanisms of activity-independent that I'm happy to talk about offline. So the very basic idea, and this is a gross simplification of activity-dependent homeostatic regulation in neural systems, is that you can use an activity sensor, and the calcium concentration in the cell is a prime candidate for that, in order for if you're the neuron, in order to assess your own current activity level and to determine whether it is at, above, or below a certain set point. And so, for example, if your set point is spiking at a certain rate and for some perturbation, your activity falls below that rate, you can sense that because your intracellular calcium concentration will be lower, because there will be fewer spikes and therefore fewer openings of voltage-gated calcium channels that allow calcium to come into the cell. And you can react to that by basically pulling all the levers and doing everything to increase your electrical activity, and that includes making any excitatory synapses stronger, making any inhibitory synapses weaker, so that will be at the synaptic level, or by putting fewer hyperpolarizing channels in your membrane to go back to your target, or more depolarizing channels in red here, and vice versa. If your activity is too high, you do the opposite. So that is a super simplified basic idea. And very early on, Larry Abbott's lab kind of did a proof of principle in this paper cited down here, and this is my reproduction of their model. So what they basically did is they had a single compartment, even simpler stomatogastric model neuron than the one that I'm using. I think it had only 5. No, it must have at least 6 membrane currents, and they implemented in this model 3 different calcium sensors that were sensing the calcium concentration at different time scales. So a sensor that would respond to very fast changes, and one to intermediate, and one to very slow changes. And then they used that sensor information to adjust the maximal conductances of these membrane currents. So there is a feedback loop, as you need for homeostasis, right? A negative feedback loop. And they hooked it up by common sense and intuition in a way such that this model now had the ability to homeostatically regulate. And here's an example of that. So what we're looking at here is the maximal conductances of these different membrane currents in this model neuron normalized to their value before a perturbation. And before that perturbation, the neuron is performing its, in this case, target activity, which is tonic spiking. And then at this case, a perturbation happens. This is a massive not massive, but rapid jump in potassium concentration, so that really throws the model out of whack. Initially, it produces non-target activity. That looks very different from what it should be doing. But then these sensors kick in and start adjusting the conductances, and you end up, after a while, you go through a couple of different activity types, and then after a while, you end up in a new state. So you have different conductances than you had before, but you're again this perturbation is, the potassium concentration changed and it stays changed. Under these new conditions, you're again able to produce your target activity. So this is kind of a proof of principle that calcium sensors can do this job. And my former postdoc, Cengiz Günay, kind of elaborated on that. And we asked, so here's just a reminder that what we're looking at here is a relatively abstract model that just posits abstract calcium sensors that eventually adjust the membrane properties. But what that really stands for is this whole cycle of regulation that we know is really going on in neurons. And I'll skip through his slides. The basic message that he found is that we were asking, what do the properties of these abstract calcium sensors need to be like such that this works, such that this thing can really distinguish functional from non-functional activity and then adjust the network properties accordingly. And it turns out that we come back to this old message that I told you about before, and that is, don't worry about the details, I don't have time to explain, but basically what Cengiz found is that the properties of these abstract mathematical calcium sensors, and by properties I mean what range of calcium concentrations do they respond to, and how quickly do they respond, described by a time constant. These properties of the sensors can vary widely, again, sometimes in terms of the time constant, over orders of magnitude, and you can still be quite successful at homeostatically regulating the network activity. So this general observation of functional activity from vastly different underlying parameters we saw now in the older work at the cellular level, at the network level. Now we see that even the sensor and regulatory systems can have widely different properties. Very elegant work that I really want to encourage everybody to read by Tim O'Leary, who I know gave a talk in this forum a week ago, but on very different work. So when he was a postdoc with Eve Marder, it always all goes back to Eve. He took the same idea, this basic idea of very abstract calcium sensors, and made it a little bit more biologically concrete, in that he really tied it to the central dogma of biology, right? Transcription, translation, and he came up with a very simple model where the calcium concentration influences levels of transcription for different types of mRNA and thereby different ion channels, and each ion channel then has its different synthesis and degradation rates. And he found that when he implements such a model that is based on really first principles and the central dogma, and uses an integral control scheme, for those of you who are familiar with that engineering term, he finds that magically he can come up with a system based on the central dogma, so biologically plausible, and an integral control mechanism that, as it's regulating to tune a model to be a pacemaking model, as it's doing that, it comes up with linear conductance correlations. So this is kind of tying it together, right? It's that if the cell is endowed with an activity-dependent homeostatic regulation mechanism that allows it to regulate its properties to stabilize proper electrical activity, that might actually be underlying this independent observation that we often see linear relationships between conductances. So I think this was really very exciting. The next two slides show experimental evidence that there is activity-dependent conductance co-regulation in the stomatogastric neurons. Again, I'll be happy to talk about that more. But I'll jump to the summary now, and then maybe we can address some of these things in the questions if you want. So I've shown that in using the stomatogastric ganglion as a model system, but this has really generalized to many other systems as well, we find that there is parameter non-uniqueness, or as people like to call it, degeneracy, meaning that the same and in particular biologically functional activity can arise from highly variable underlying parameters. I have robustness in the title of the talk because if you think about this, I didn't point this out earlier, that means that's good news for biology, right? It means that a system doesn't have to tune itself to one unique parameter set in order to function properly, but it has a whole solution space at its disposal. And so that also means that if it sits not at the edge of the parameters of the solution space, but kind of smack in the middle of it, it's pretty robust to perturbations, because it'll be perturbed out of where it's sitting, but it'll still be able to produce functional activity. So that what I just said also reminds you of the notion of solution spaces. We have seen that they can have pretty complex structure. I only showed you implicitly, but we do think that they are generally connected in parameter space. Often these solution spaces show parameter correlations, and we've seen that in electrophysiology, in mRNA copy numbers for ion channels, and in models. And I've briefly touched on mechanisms of activity-dependent regulation of these parameters and these correlations in the spirit of homeostatic regulation. And I just want to again point out that there are many putative homeostatic regulation mechanisms, only some of which are activity dependent. There are also activity independent mechanisms that I didn't even touch on today. So that takes me to my last slide, which is thank you slide. These are three members of former members of my lab that whose work I showed: Amber Hudson, now Amber Willett, Cengiz Günay, and Quinn Tian. I also briefly showed the dimensional stacking approach that was developed with the Hickey lab and Joan Langton in particular. And above all, I have to thank Eve Marder, who, like, even 15, 16 years after I left her lab is still, I wouldn't be where I am without her. So she was really a major force in my development as a computational neuroscientist. And funding sources. And I'd also like to thank you for your attention, even though it's in an online setting like this. I can't even see anybody's faces. It's a little bit anonymous, but thank you for taking the time. And thanks, Tim, again for inviting me. This is it.
Thank you very much, Astrid. I was just looking for the link to the Zoom room, so...
oh, right. So now we go straight into the Zoom room, right?
Well, no, actually, we have a few people here that have questions. I already have Peter Jonas on screen, so I'm going to unmute his microphone and give him the floor for a sec while I see if I can invite a few others.
Should I continue my share so that I can flip to slides?
Yeah, I can minimize for now, and then if you have a question, Peter, why don't you go ahead?
Thank you very much, Astrid, for the nice and stimulating talk. I was wondering about the importance of channel gating versus conductance, maximal conductance, And I was wondering how important channel gating is in your solution space, and whether there's a similar degeneracy for channel gating as you have shown for maximal conductance for the individual channels.
Yes. So as you noted, in our work, we focused on varying the maximal conductances only, and we kept everything else constant. That is, gating variables, like the voltage dependence of gating variables, time constants of activation and inactivation, and that is motivated by two factors. One, that with this brute force approach, of course, every additional parameter that you vary kind of explodes the number of computations that you have to do. But then a more biologically motivated reason is that maximal conductances do appear to be more variable than gating dynamics, gating properties. That's not to say that those can't also vary, and we definitely see that that in different cells you don't always measure the exact same half activation voltage, and also, of course, different splice variants can have different properties. So if we're thinking about how is all of this regulated, there are many ways where regulation could happen, many stages where regulation could happen on the way from a gene to a functional ion channel, right? And post-transcriptional changes, for example. So absolutely, the gating voltage dependencies and dynamics are not rock solid, exactly the same in every cell, but they vary, to my knowledge at least, they vary less than maximal conductances, and so we chose to not vary them in this approach. Other people have started playing around and doing explorations, I believe, including some of the gating dynamics, and also people have started using this approach of ensemble modeling and trying to look at different constellations, even at the level of morphology, of cell morphology, looking at if I have an ensemble of cells that have statistically the similar dendritic trees, but they vary in the details, what does that tell me about general principles of signal processing? Maybe I veered away from your question a little bit. I hope I answered it.
It's fine. Thanks a lot. We talk later.
Okay.
Thank you, Astrid. Steve, you had a question too. Why don't you?
Yeah, you guys hear me?
Yeah.
Yes.
Hey.
Hi.
Hello. Long time no see.
Yeah.
So I guess my question was about, on one of your early slides, you were comparing the variability of different parameters in the simulations that you ran and the variability that you saw in experimental results. And I don't know how directly comparable those two are. On that specific slide, they were in different units, and I know the models don't always correspond directly with reality, but I guess I'm wondering, of that big 8-dimensional space you saw, if there's some big space where you see bursting that looks like biological bursting, how much of that space is filled with like actual experimental results? Like how many of those models have like a real cell that's nearby them?
That's a very good question, and disappointingly, but maybe interestingly, the answer is that the model solution spaces that we find, even for like a stomatogastric neuron, and when we compare it to stomatogastric data, don't necessarily always match what's seen in biology. And that was the one slide where I said it's complicated. That would have kind of made that point, but it would have taken me like 10 minutes to explain it. So what we find is we find basically all combinations. We find correlations that exist in biology and that we see in the models. So those are kind of reassuring. We find correlations that exist in the models but not in biology, and vice versa. And maybe most interestingly, we also find that the models, they produce a lot of positive correlations, where one conductance increases and the other also increases, so a positive slope. but the models also produce quite a few negative correlations. But in biology, those are very rare. Actually, for years, I was kind of challenging people at the end of my talk to bring me any kind of observation where they saw a negative correlation. And to me, that is very interesting because, you know, not being a biochemist or a molecular biologist, naively, I would have said from the electrophysiology perspective, if you have two calcium currents like these models do, they have a transient and a slow calcium current. These calcium currents differ in their dynamics. That gets back to the previous question. But they're still both calcium currents. So you might imagine that if one of them is big, the other shouldn't also be big. That you want to kind of maintain an overall level of calcium influx. But that is... So that would suggest a negative relationship. And from biology, I understand that most co-regulations rely on a transcription factor being upregulated, and that it then increases the expression levels of two different things, and so they would kind of naturally increase and decrease together. But in principle, some molecular biologists tell me in principle you could also implement a transcription factor that downregulates one thing and upregulates another thing, and that would again give you a negative relationship. So your question was more general about how does biology actually exploit the whole model predicted solution space? And my long-winded answer that again veered off topic is there's not a perfect match between the two, but I think that's always then the challenge is what can we learn from that?
Yeah, and the point about the correlations is interesting. I feel like that tells us something about the sort of control mechanisms that biology favors over what's theoretically plausible.
Right, and we also, one thing that my former postdoc Tomasz Smolinski explored is, so in my original model neuron database, you know, I pointed out that each conductance can vary from 0 to some physiologically reasonable value with some increments in between. And with Tomasz, what we were asking is, what if we allow the conductances to vary freely, like even to insane levels way beyond the physiological range? And he ran a multi-objective evolutionary algorithm and created a population of models, of stomatogastric model neurons that was not constrained by the physiological range. And he found that he could find these linear relationships extending to like 100-fold beyond the physiological range. So the model would say, this solution is just fine, you know, increase these two things 100-fold, you'll still be doing a good job. And I think that's a good example to make the point that I think we need to apply many more constraints to these models. And one obvious constraint that I'm looking forward to talking to one of Tim's postdocs about is metabolic cost, right? Sure, you can do the same job with 100-fold of current flowing in and out, but that will cost you energetically, which we have not applied a metabolic constraint to our models yet. So I think once we start making the models and the model ensembles in particular more realistic in the sense that we don't just that we think about additional constraints, hopefully the shape of the model space, solution space will slowly kind of converge on the actual shape of the biological. And one kind of technical issue is so at the mRNA level, Dave Schultz, who is mostly doing that work, can now measure pretty much the entire complement of ion channels in a single cell, of mRNA in a single cell. So there you have at least those dimensions, you have the whole information for a cell, but electrophysiologically, you can usually not measure all maximal conductances. It's just, you know, washing in and out various toxins that may or may not even be specific in that system. So it's experimentally, it's hard to get a real grasp on what is the real solution space.
Astrid, we have two more questions from the audience before we maybe swap into the Zoom room. Steve, sorry.
Okay.
No, that's fine. Thank you very much.
Yeah, good to see you, Steve. And you're
feel free to shut your cameras if you want to, but you're welcome to stay online if you want. If you have follow-up questions. There was two questions from the audience. One was, do you think...
Are we switching to the Zoom room or not?
Well, let me quickly ask those two questions and then
okay.
Number one was, do you think there are homeostatic mechanisms in place for neurons that don't fire spontaneously? And if so, what are they? And are there neurons that don't fire spontaneously in your circuits?
Yes. Okay. So that's a interesting question, and it kind of gets back to when I first explained the Pyloric circuit. I said that there is the pacemaker kernel which is intrinsically bursting, so it produces bursts in the absence of synaptic inputs. But the other two types of neurons, the LP and PY, are also often called follower neurons, in the sense that if you synaptically isolate them, if they don't get that periodic inhibition from the pacemaker, they can be either tonically spiking, or sometimes they can be silent. This varies from animal to animal. And so there you have an example of a silent neuron, of an intrinsically silent neuron. It's not by itself producing electrical activity. It does burst in rebound from inhibition, from periodic inhibition from the pacemaker. But there you have I'm not sure if that's what the question asker was thinking about. There you have an example of a silent neuron, and now the question is, does it still undergo homeostatic regulation?
Yes.
And well, at least I can say that these follower neurons that are not intrinsically active, they do also show their own correlation rules. Whether that means that they necessarily undergo homeostatic regulation to maintain activity-dependent homeostatic regulation, that to maintain those co-regulation rules, I'd have to think about whether there's any evidence for or against that. I mean, Tim O'Leary's work that I briefly talked about shows that in principle, the central dogma, together with a simple integral feedback scheme, can produce calcium-dependent and therefore activity-dependent correlation rules and homeostasis. And I mean, maybe this is a partial answer to this. In a sense, if you have a homeostatic regulation rule that depends on activity, that mechanism will engage no matter what causes that activity, right? Whether that activity is produced intrinsically by the cell or whether it's in response to inhibition and rebound. So calcium influx is calcium influx in some sense, but not in other senses. I'm not sure, did I miss the question?
No, I mean, I guess it's difficult to imagine a neuron unless it's constantly on the brakes and prevents itself from firing completely if homeostasis is only going towards silence, right? It's somewhat difficult to imagine a mechanism that would homeostatically arrange itself so that it can still respond. I guess there must be, maybe not difficult to imagine, I don't know, that arranges its ionic conductances such that it will respond to inhibitory rebound, but not fire otherwise. I don't know.
Well, they do exist. Like I said, there are some of these follower neurons are silent and they only burst if they get that periodic inhibition. But then, you know, once they burst, they have that calcium influx and they can use that as a mechanism to adjust themselves.
Yeah.
And also, we haven't even touched on synaptic plasticity and all of that, right? So that is also a possibility.
Anand said his question was answered, so I guess he gave us
Okay, good. Thank you.
And the other question he had was, why not quantify the expression levels of a particular ion channel instead of looking at mRNA level, as the mRNA levels and protein levels are not always correlated?
Maybe you were talking a little too fast. I didn't catch the first part. What about the?
Why not look at the expression levels of particular ion channels instead of looking at mRNA levels, as the mRNA levels and the protein levels are not always correlated?
Yes. So there is work by Dave Schultz, who is the man doing the single cell mRNA work, together with Jean-Marc Goyard, who was also an overlapping postdoc in the Marder lab. And what they did is, Jean-Marc, who is the electrophysiologist, would identify a neuron in the ganglion, measure with electrophysiology, measure as many of the maximal conductances as he could. And as I said earlier, you cannot measure the full complement electrophysiologically, but he would measure as many as he could. And then they would pull that exact same neuron, give it to Dave, and he would do his mRNA copy number analysis on that same neuron, and then they would compare conductance levels, which corresponds to functional ion channel protein, and mRNA copy number. And there again, the news is not as simple as one would hope, but therefore again maybe interesting, and that is I think for about 2/3 of the ion channel types, they found a positive correlation between mRNA copy number and protein and maximal conductance, which is kind of a proxy for protein expression levels. But for the rest of them, they were not correlated. I think there was even one case where the mRNA copy number was negatively correlated with the amount of conductance. So you were asking about protein expression levels, and because I'm not. Yeah.
If mRNA levels and protein expression were always on par, or whether mRNA was a true measure of protein or channel density, essentially.
But there's also here's another kind of complicating story in that respect, and that is early work from Ron Harris-Warwick's lab at Cornell when he was still working in the stomatogastric ganglion, and this was in the late 1990s, I believe. So what he did is he was just generally interested in how does cellular activity depend on the expression of different ion channels, and so he overexpressed one of the potassium channels, the A-type potassium current channels. He overexpressed that in a stomatogastric neuron, and the plan was to just look at what does that do to electrical activity. And he found that he could show that the channel protein was expressed and inserted into the membrane and was actively conducting current, because the A current magnitude went up dramatically when he overexpressed. But the electrical activity, the bursting activity, did not change at all. And so what he then found out is that what happens is, in response to this upregulation, this artificial upregulation of the A-type potassium channels, the cell responded to that with a compensatory upregulation of the hyperpolarization activated cation channels. So why did I launch into this story? Now I have to remind myself. So that means that and then in a follow-up paper, now he overexpressed that same A-type channel, but a mutant of it he could show that was expressed, inserted into the membrane, but did not conduct any current. But the cell still responded by upregulating its H-type current. So that is an example of basically his argument there was, and even I wrote a little commentary on it in they published this in Neuron. His interpretation was the cell upregulates its H-type channel not because of an activity-dependent regulation, because the activity didn't change, but because of the mere presence of the A-type channel protein or mRNA. Like I think it's still not clear at what level of the cascade this interaction between A and H happens. Again, I tend to get triggered by a question and then launch into a story that may not necessarily be right on target. But
Astrid, I think we have to stop here. In fact, I think we ran out of time for the post-Zoom room, post-talk Zoom, and I'll have to usher you straight into your next meeting with students and postdocs. So I'd like to say thank you again for coming, and I will send you the next link by email for this.
On my schedule that I got, the meeting with students and postdocs is at 11.
Is that not right now? Hold on, let me check. Well, then we have a little more time. Then we can go to the post-talk.
Yeah, I mean, I don't.
Okay, so then let's go into the post-meeting room and see if you have any takers still. So I'll see you there.
I close this and then I go.
Yeah, you can even do it at the same time. You can just click on the Zoom room link in your program, and I'll see you on the other side right after this.
Zoom room link in my program. Where is that?
In the program that Jessica sent you, or in the chat here.
Oh, in the schedule. Yeah, that's what I was going to do. Yeah. Okay. So.
All right. So for now, goodbye, and see you on the other side.
Okay. So then this broadcast is done. Yeah.
That's right.
Okay. Bye.